Professor Clemens Kunz from the Institute of Nutritional Sciences at the University of Gießen in Germany spoke about the latest research on human milk oligosaccharides, or HMOs, during the NNI Symposium, which was held at the 3rd International Symposium of Prebiotics and Probiotics in Pediatrics in Ghent, Belgium in 2016. Professor Kunz explained why HMOs are not just sugars but could be nature's secret weapon. He also highlighted the findings from various clinical studies indicating an association of HMO profiles and the risk of certain diseases. According to Professor Kunz, HMOs represent the largest compositional gap between the human breast milk and infant formulae. HMOs have unique structures with special functions and undergo a specific metabolism. That's why HMOs cannot be compared with commonly used prebiotics. Moreover, Professor Kunz expects the advent of a new research era for investigating the role of specific HMOs in clinical studies as a result of the enormous progress in producing HMOs at a large scale. Most of the HMOs consumed reach the colon, where they are utilized by the gut microbiota, and thereby the growth of beneficial bacteria is promoted. Further, HMOs prevent pathogen adhesion in the gastrointestinal tract and contribute to gut maturation. Only a small percentage of HMOs are absorbed and reach the systemic circulation, suggesting additional tissue functions. Recent studies suggest that HMOs have anti-inflammatory and anti-infective properties, and they may also influence the brain function of infants. One of the major factors influencing the composition of HMOs in human breast milk is the mother's secretor status. About 80% of the population belong to the secretor group and express the enzyme fucosyltransferase 2, or FUT2. The predominant HMOs of the secretor group are 2-fucosylactose, or 2FL, and lacto-N-fucopentaos, or LNFP1. The remaining 20% belong to the non-secretor group, who express the enzyme fucosyltransferase 3, or FUT3. The mothers in the non-secretor group secrete only very small amounts of 2FL and LNFP1. Professor Kunz further presented interesting data indicating an effect of the secretor status or specific HMO profiles on the risk of certain diseases. For example, a study by Morrow and colleagues found that an abundance of 2FL is associated with a reduction of Campylobacter diarrhea in infants. McGovern and colleagues suggested that the mother's non-secretor status is associated with a higher risk of developing Crohn's disease. In another study, Morrow and colleagues showed an association between low or non-secretor status and an increased risk of mortality, gram-negative sepsis, and necrotizing enterocolitis in preterm infants. Lewis and colleagues demonstrated that the microbiota composition in infants is dependent on the FUT2 status of the mother. Breastfed infants of secretor mothers had higher levels of beneficial bifidobacteria and lower levels of streptococci in the gut than infants of non-secretor mothers. In his outlook, Professor Kunz proposed that the large-scale production of smaller HMOs will stimulate further investigations into HMO benefits for infants. Human or animal studies should further examine the structure-function relationship of specific HMOs and the effects of HMOs on different gut microbiota, not only on bifidobacteria. Moreover, a better understanding of the genetic influence on HMO profiles and their effect on babies' health is needed.